Good day, everyone. I am Hazel Faith Galantes, and today I will be discussing the E.Coli chromosome map. This map shows how the genes of E.Coli, a commonly studied bacteria, are arranged on its circular DNA molecule. Understanding this map is important because it helps scientists locate genes, studies DNA replication, and perform genetic engineering. Unlike humans who have linear chromosomes, E. coli has a single circular chromosome made of double-stranded DNA. It contains about 4.6 million base pairs and around 4,000 genes. The circular shape is what allows bacteria to replicate DNA very efficiently. The map places the replication origin called REC at the top of the circle. This is where DNA replication begins. Specific DNA sequences at REC bind the protein DNA, which unwinds the DNA double helix. You can think of REC as the starting line for DNA replication. From REC, replication moves in two opposite directions around the circle. On the map, this is shown by two large arrows, one going clockwise and one going counterclockwise. This is called bidirectional replication, and it allows the interchromosomes to be copied faster. Halfway around the circle, opposite REC is the terminus region. This contains TER sites like TER A and TER C, and a special site called diff. These sites stop the replication forks so that the replications end properly. Diff also helps ensure that duplicated chromosomes separate correctly during cell division. In simple terms, this area is the finish line. The map also highlights several rRN operands labeled rRNA, rRNB, rRNC, and so on. These are clusters of ribosomal RNA genes. They are highly active because E. coli needs ribosomes to grow and divide quickly. Their placement on the map helps researchers recognize key gene clusters. Other labels on the map show gene names followed by numbers. For example, ILES 22420. The number indicates the exact base pair position on the chromosome. The color of the label shows which DNA strand the gene is located on. This makes it easy for researchers to locate and work with specific genes. So, why do we make chromosomes maps like this? Chromosome maps allow scientists to locate genes precisely, design genetic experiments, and understand how DNA replication interacts with gene expression. In biotechnology, this helps when modifying E. coli for useful purposes such as producing insulins or enzymes. Chromosome maps are also valuable for research on antibiotics and for comparing harmless strains to disease-causing ones. To summarize, the E. coli chromosome map gives us a visual guide of where genes are located and how DNA replication works. Replication starts at REC, moves in two different directions, and ends at the terminus. This map is an essential tool for microbiology, biotechnology, and genetic engineering. That will be all. Thank you.